Now we will quickly move on to the third subtopic, which is formation of heart tube. So for this subtopic, we will once again draw the larger picture of trophoblast with what is called as the epiblast with its cavity amniotic sac. And then we have the hypoblast with its cavity, which is yolk sac. And we have what is called as the primitive streak notochord and the newly formed horseshoe shaped heart field at its cranial end. So, this picture, what we are seeing exactly at the level of the primitive node or the primitive streak. Suppose this could be seen at a different level, which is exactly at the level of the mesoderm, and another cross section view. So we have what is called as the primitive streak with notochord. Cranial to that is the developing horseshoe shaped area of cell, which is the cardiogenic region. And we have what is called as the mesoderm, which is entirely formed laterally, covering even the cardiogenic region. But this mesoderm is absent at two regions of the embryo where this epiblast and hypoblast are meeting each other. So if you could imagine epiblast, we have this hypoblast. Now this epiblast and hypoblast initially together, now separated by a sheath of mesoderm in between. But this mesoderm is not available at two regions of embryo, one at its cranial end, which is called as this buccopharyngeal membrane, and its lower caudal end, which is called as the cloacal membrane, which is going to form the future anus. Remember, in initial stage, the buccopharyngeal membrane is caudal to the cardiogenic region, or in other words, the cardiogenic region is cranially placed to the buccopharyngeal membrane. So this is one part of diagram. If I put this in a bigger picture, for instance, if I draw this at this, make it into this diagram. So we are seeing this diagram exactly at the midline. So we are seeing what is called as the primitive streak. Suppose we take the primitive streak, we cut the embryo at the level little laterally so that we can get the mesoderm into the picture. So we will get what is called as this kind of picture. Epiblast with extra embryonic mesoderm, hypoblast with extra embryonic mesoderm of hypoblast. We have the sheath of mesoderm in between, except at the caudal and the cranial end, where these two structures are going to meet each other. They are going to kiss each other like this. Like this. So and Here we have what is called as the horseshoe shaped structure of heart field. So this extra embryonic mesoderm is here. Whereas this mesoderm is covering, the lateral plate mesoderm is covering even the cardiogenic region part, but it is not covering this end. So this is covering the cardiogenic meso or cardiogenic region. So this is the extra embryonic part. So this mesoderm it laterally, this is the most lateral part of the mesoderm. Exactly the lateral part is covering. So here you can see exactly the lateral part is covering this end of the heart field. So here at this cross section, the same thing, it is covering the lateral part is covering the cardiogenic region. So this lateral plate mesoderm, which is covering this region, gets divided into two. One is the somatopleuric mesoderm and the splanchnopluric mesoderm. So this somatopleuric mesoderm gets continuous with that of the extra embryonic mesoderm of epiblast. So this is somatopleuric mesoderm getting combined or continuous with that of the extra embryonic mesoderm of epiblast. And similarly, the lateral plate mesoderm, so this is the lateral plate mesoderm here, which gets combined with that of the extraembryonic mesoderm of hypoblast is called as splanchnopluric mesoderm. So this is the extraembryonic mesoderm of hypoblast. So we will draw this, we'll combine this the same on either side. Try to combine this. This is the area where they are meeting with each other is the cloacal membrane at the caudal end, and then this entire extra embryonic mesoderm is now covered by this both the splanchnopluric and the somatopleuric mesoderm. So this gets covered like this. So now this region of embryo where the heart is developing is now called as cardiac mesoderm. Remember, at initial stages, the cardiac mesoderm or the developing cardiogenic region is cranial to the embryo, or in other words, cranial to the buccopharyngeal membrane. So this development of the heart field and the cardiogenic region happens during the middle of third week. So the development of heart field has started at the middle of the third week. So initially, the cardiogenic region, which is having the horseshoe shaped cells, it's an island of cells which is forming this structure, arranging themselves into this structure. So this blood of islands, they actually start developing into thin endothelial lined tubes. So initially it is a single tube. Later it forms into another tube. So two separate endothelial lined tubes is what is the initial formation of heart. So now we can see a bigger picture of this, of a developing embryo. So we can name these parts, easily we can name these parts, right? So this is the developing embryo epiblast. This is the hypoblast. This is the primitive yolk sac. This is the amniotic cavity. Here we have in between the green structure, the mesoderm. And then we have the cardiogenic mesoderm or cardiac mesoderm developing the two endothelial heart tubes. And then they are lined by the lateral plate mesoderm. Above we have the somatopleuric mesoderm. Below we have the splanchnic mesoderm, which is engulfing the both the extra embryonic mesoderm and this area or region where the epiblast and hypoblast is not covered or not separated by the green colored mesoderm. This area is the buccopharyngeal membrane. This is the cloacal membrane. Right. So initially the heart which is developing in this region is cranial to this buccopharyngeal membrane. So as the embryo is growing, this embryo is going to engulf and it's going to fold both cranially it starts growing and laterally it starts growing, and somewhat we get this kind of picture. The same thing, nothing is changed. Here you have what the one change that has happened because of this growing embryo growing, enlarging, and because of that the cranial structure, the cardiac mesoderm, which is having the growing heart, is now pushed into a more caudal part or into the thorax. So here you have this heart now placed. Also, the primitive yolk sac is now pushed into the developing embryo, and this now forms what is called as the secondary yolk sac. So once again, developing embryo from the ectodermal cell layer, we have the mesoderm, we have the hypoblast or the secondary yolk sac layer, which is giving rise to the endoderm. Here, this area where you have the both yellow and the blue region, which is seen together, is called as buccopharyngeal membrane. Here, once again, this region, yellow and blue together, cloacal membrane. This is the amniotic cavity where the embryo is developing. This is the secondary yolk sac, and this is the somatopleuric and splanchnopluric mesoderm, from where we have the connecting stalk. So now the heart is pushed into the thorax. And here you can see this buccopharyngeal membrane is becoming more cranial. Before it was caudal to the cardiogenic region. Now, because of the embryo folding cranially as well as laterally, it has pushed more cranial, and the cranial cardiogenic region is pushed caudal and now gets to lie in the thorax. Now we will concentrate on our cardiogenic region alone. So here, as I told you, the heart tube is developing inside this cardiogenic region or cardiac mesoderm. So this cardiac mesoderm has two layers of mesoderm. One is the somatopleuric mesoderm. Second is what is called as the splanchnopluric mesoderm. So you have a somatopleuric mesoderm and this is splanchnopluric mesoderm. So the cavity which is formed is now termed as the pericardial cavity. So we now come to know that pericardial cavity is lined by both the somatopleuric mesoderm and splanchnopluric mesoderm. And this heart tube is actually suspended. It's not floating inside the pericardial cavity, but it is suspended by this mesoderm. In other words, it is suspended by the mesoderm dorsally. Thereby, it is called as dorsal mesoderm. And the pericardial cavity has this roof. So somatopleuric mesoderm forms the roof of the pericardial cavity and the splanchnopluric mesoderm forms the floor of the pericardial cavity. In other words, in future, this roof or the somatopleuric mesoderm forms what is called as the parietal layer of pericardium, and the splanchnopluric mesoderm forms the visceral layer, in other words, the epicardium. I once again explain. The heart is a tube. Two endothelial lined tubes are suspended in this cavity of cardiac mesoderm by means of dorsal mesoderm. So it is not attached ventrally. And this cavity is called as pericardial cavity, which we all know is guarded by both somatopleuric as well as splanchnopluric, where the somatopleuric mesoderm forms the roof of the cavity and splanchnopluric mesoderm forms the floor of the cavity. And this roof gives rise to the parietal pericardium. The parietal part of pericardium, if you know the anatomy, has two parts. One, it is the fibrous and the serous part of pericardium. Whereas the floor or the splanchnopluric mesoderm gives rise to the visceral pericardial layer, in other words, which is called as the epicardium. Not only the splanchnopluric mesoderm give rise to the visceral or epicardium, it also gives rise to a structure called as myoepicardial mantle. Myoepicardial mantle, which in future will give rise to what is called as myocardium. So with that, we finish off our third subtopic, formation of heart tube. Hope you understand.